Good morning, good afternoon, and good evening, and welcome to Chapter 8, Cellular Reproduction. Today we're going to talk about how cells reproduce. Obviously, cells reproduce by dividing in half. There are three processes that cells use to divide, depending on the kind of cell that they are. The purpose of this division is to replicate the cell. One cell will divide in two. One cell will become two cells. Two new cells that will then live and go on at some point at the end of their lives to replicate and divide on their own. This one beginning cell is called the parent cell, sometimes called the mother cell, but usually called the parent cell. That parent cell will split in half. Usually dividing the bits and pieces of the cell equally amongst the two resulting daughter cells. The daughter cells are the new cells that are going to pick up the torch and carry it to the next generation. Before the before the one cell can split in two, the cell has to replicate its chromosomes. If we're talking about prokaryotes, there's just a single chromosome. We'll talk about how that replicates when we get there. If it's a eukaryote, it's likely to have multiple chromosomes, and it's going to have to duplicate those before it starts. Please try to think about this and remember, imagine that a complete set of chromosomes just is $1, right? So before one cell splits in two, for those two cells that we're going to get at the end, for them both to have a complete set of chromosomes, what did the original cell have to have before it began the process? For each of the resulting cells to have a buck in them each, a complete set each, that means the beginning cell had to have had two complete sets inside of it. The beginning cell had to have $2 in it, so that when it splits in half, it will result in two cells that have $1 each, two cells that have a complete set each. So before the dividing process happens, a eukaryotic cell is going to have to replicate its DNA. It's going to have to copy its chromosomes. And we talked about this a long time ago, and we said, you know what, I want you to think about chromosomes like recipe books. And a recipe book has recipes in it. Chromosomes have genes on them. And you have a certain number of them, and that's what makes you a human, or makes you a pea plant, or makes you a gorilla, is your DNA, your chromosomes. And if we imagine this parent cell as having a complete set of recipe books, before this parent cell dies, if this parent cell wants to be sure that both of its daughters have a complete set, what does mom have to do to that set of recipes? What does she have to do to that set of books before she dies? She has one set. What does she need to do to that one set before she dies to make sure that both of her daughters each get a set? And the answer is she has to make a copy of it. She has to make a copy of that set. So right before she begins the dividing process, right before she dies, she doesn't just have one set, complete set. She has two complete sets. Cells will divide the sets of chromosomes equally between the two daughter cells, and that's what we just said, so that each daughter cell has a complete set. There are two methods by which cells will reproduce, by which organisms will reproduce. The oldest and simplest method is asexual reproduction. That is, an organism will simply just decide it's time to make the next generation, and it will just start to divide. The offspring are genetically identical to the parent because there is no mixing of DNA. This is cheap. This is easy. You don't need a partner to do this. You don't have to wait for a certain time necessarily to do it. You just do it whenever you're ready to do it. It is the most common, but the problem is that all of the genes in the next generation belong to a single parent. And so if that one parent has some sort of a genetic weakness, then all of the babies will have it too. And if bad circumstances arise that take advantage of that genetic weakness, well then all of them will die. Asexual reproduction. The other variety is sexual reproduction, and that is where two separate individuals will each contribute DNA to the next generation. And what this does is this mixes up the DNA. This gives you a new combination of genes, a new combination of instructions, hopefully the next generation won't have whatever the genetic weakness was in the first generation, because hopefully when I give my DNA to my wife, hopefully the baby will get the better combinations of DNA than I have or she has, mixing up the DNA to make brand new individuals with the hope that they will have a better combination, a better set of DNA that gives them the ability to live longer, live in more varied environments, and to do better, to do well. That's the whole advantage, that's the whole purpose behind sexual reproduction, to mix up the DNA with the hopes that the next generation will be healthier than the previous generation. There are some problems with this, though. The first problem is you have to find a partner to do sexual reproduction. That's not always easy. The second is it can be dangerous. We all know there's a whole bunch of sexually transmitted diseases you can get simply by having sex with someone, so that's a danger. It's also dangerous in that a lot of times individuals will compete for mates. Very often males will fight one another for access to females, and so again, that's a danger. So sexual reproduction, it is different. It's arguably better than asexual, but that doesn't mean that there's not problems that come with sexual reproduction. There are problems that come with it, as we already talked about. The possibility for disease, the possibility for danger. Not being able to find a partner is a problem. It happens. It does happen. All right. So before we get too much further into discussing cellular reproduction, we need to talk a little bit, remind ourselves a little bit about what we call cell theory. And this is that new cells come from existing cells. We talked about this before. You know, that moment your life began, you were one single cell. What was that one single cell 10 minutes before that? That one single cell was a sperm and an egg, right? Your mom's egg and your dad's sperm cell. And if we take, either of those cells, the sperm cell, we carry it back. How far back can we actually carry that sperm cell? We can carry that sperm cell all the way back. The origin of that sperm cell all the way back to where dad was a single cell inside of his mother. And then what was that single cell? And just keep going back and back and back. New cells come from existing cells. Life does not spring from nothing. It has to come from life. Life makes life. The simplest form of reproduction is called binary fission. Binary means two. Fission just means to split, to crack in half. So binary fission literally means to split in two. Bacteria have a single circular chromosome. And remember that DNA is like a ladder, all right? Well, bacteria have circular chromosomes, so now you have to turn a ladder into a circle. So the ladder, the circular chromosome of bacteria, you can actually kind of imagine it similar to a hamster wheel. Have you seen like those metal hamster wheels that, it's got to kind of stretch it a little bit to make it look like a ladder, but it is essentially just a ladder in a circle. So the DNA, the chromosome of a bacterium, is a circle. And what you're going to do is the chromosome is going to attach to the surface, the inner surface of the cell wall. And then as the bacterium splits in half, it's going to split its chromosome in half. And again, we're going to take that hamster wheel and going to cut it into a left and right side so that the spokes are sticking out like this. Okay? All right, and then you have the other side. Okay? Two halves of my hamster wheel, all right, that have been cut in half. This is called binary fission. Here we see our bacterial cell with its single circular chromosome, and I know it doesn't look like a circle to you, but just imagine like a big, a huge rubber band all wadded up into a chunk. That one circular chromosome will attach to the cell surface. When it does, the cell will begin to split right there, and when it does, it splits the chromosome down the middle into the left and right half. And because of how DNA works, the way it works is if I have an A on this side, I know what's supposed to go on the other side. I put a T. So once I have split my hamster wheel in half, I can remember, I can make that other side. I don't need to have both sides to be able to replicate it. So I can take my one chromosome, split it in half, and end up with two of the very same chromosome, each one being half old and half new in the new cells. This is the process called binary fission. One bacterial cell ends up splitting in two.